Hi, welcome to our short movie for the new drug of pulmonary fibrosis. I'm Yasuo Saki, had been a professor in Hiroshima University and from Antibody Therapeutics Incorporation today. Our team has been interested in cell matrix whispering with occasional flare-up. The problem we show is IPF having huge unmet medical needs. There are no curative treatments except transplantation. Patients notice the terminal while drowning on bed day by day. How hopeless and unfair. When I was in charge of the patient with IPF, at the time she passed away, I thought how incompetent we physicians are. Although there are two approved drugs, neither halt disease nor improve QOL. A new treatment option is eagerly awaited. Globally, 100 to 200 million people are suffering. Recent findings suggest coming discovery of the curative drugs. We found antibody that binds to fibroblasts via an integrin and inhibits activation through two different mechanisms. First, as in picture on the right top, fibroblasts plated on the specific ligand for the integrin, nephronectin, shows stress fiber formation, a hallmark of the activation, indicating our target integrin mediates input of signal for the activation. Second, the target integrin activates latent TGF-beta. As in the plots, respiratory reporter cells sensing TGF-beta signal emits higher luminescence when co-cultured with lung fibroblasts, which is reduced by knocking down target integrin expression. Encouraged by these results, next we examined the effect in 6 different animal fibrosis models and found elevation of fibrosis in each model in the lung, liver, kidney, and gingiva. Fibrotic markers were reduced in the lung, and the fibrotic area narrowed in the pictures in the liver and gingiva. The target of the antibody is integrin alpha 8 beta 1, which has 2 important characteristics suitable for the target. Its expression is highly specific to fibroblasts and is rapidly and markedly upregulated upon fibroblast activation. Thereby, it's abundant in the fibrosis but not in normal tissues. Every antifibrotic candidate drugs are said effects are through fibroblasts, but so far no drug solely targets those on the fibroblast except ATI-235, our antibody. Of note, the authentic key player for the TGF-beta activation is integrin alpha-V beta-6. Alpha-V beta-6 is expressed on alveolar type 1 cells, and importantly, alpha-8 beta-1 on fibroblasts. As the disease progresses, alpha-V beta-6 played the role before alveolar destruction. Then fibroblasts intrude into the space. Alpha-8 beta-1 is there on the fibroblast. This could be a critical sequence to understand the drug mechanism. In clinical trials, many expected drugs have been terminated. Why so many have been terminated? Animal fibrosis are developed in non-physiological severe setting. To overcome the given model's specific huge fibrogenic power, considerably potent drugs have been selected. That's my guess.
The IPF market is characterized with a clear unmet medical need and as a commercially attractive market. Therefore, the optimal scenario of developing ATI-235 fast in the global market is based on an out-licensing business model at IND after ATI prepares a comprehensive preclinical data package. We hope the licensee is a multinational pharma company who accelerates the clinical development of the product through global phase 3 studies. With a successful launch, the deal value can be significant with an attractive royalty stream. The challenge, however, is in the preparation of a preclinical data package which is convincing enough about the differentiation. ATI has an idea for such a data package and needs funds for the data creation. The initial target of ATI-235 is IPF, which can be designated as orphan drug. In Japan, the current IPF market size is $430 million US, and in the global market, over 3.3 million. Talking about the peak sales estimation of ATI-235 in Japan, the sales can reach 300 million. This is ATI's roadmap showing the growth scenario. We have not only ATI-235, but also other assets created from integrin research. One of them has been evaluated under an MTA by a global top 10 pharma. The scenario is associated with a series of out-licensing of ATI products to earn milestone payments, then followed by an IPO. The IPO should enable ATI to continue its integrin research to create next druggable assets. The outlook is bright, assuming successes of the three assets. In our expectation, we'll have two out-licensing opportunities with ATI-235. The first one is at the IND, and the second one with completed phase 1 data conducted by ourselves. To make it happen, we really need $5.7 million US by the end of 2025 and possibly additional $5.1 million US from 26 to 2027.
This is our team slide. We are joined by 3 special persons last year for business. Thank you for attention.